Well, this is a really important lecture because actually one of the most important jobs that master gardeners do is we're sort of amateur or learning, budding plant pathologists. I mean, really, we have to do a lot of diagnoses on the helpline. So this is a really important lecture.
Well, and Brian did mention that to me. He said, the better you help train the master gardeners, the more they can answer the questions for you. And then something that's very much out of the ordinary, they will bring it to you. And so when Brian retired, there was a list of how many new introductions that he had found over the 30 years that he worked. And, it was more than a page. So all of these things that are seen for the first time, you have to recognize what's already here or what is new and different. And it's literally usually going to be the master gardeners that are in touch with more people. If you want to have a plant pathology analogy, mycorrhizal fungi, they are fungi that live in association with plant roots. So mycor is for fungi, rhizae is for roots. And in general, it helps both the fungus and the plant. The fungus, of course, gets sugar from the plant that the plant makes from sunlight with carbon dioxide. But what does the plant get from the fungus? It gets a much wider network. And so, you guys are the fungus mycorrhizae, and, I could be the plant person getting the information. I'll give you sugar. I'll give you sugar. You give me some phosphorus, I give you sugar, and we're all happy. And one of the things in this analogy that doesn't always work down to the nth degree is that it's like they're both trying to help each other. It's like not actually. If you have a tree seedling that is shaded or something, the fungal micro then you have a pathogenic relationship, which is one is harmed and the other benefits. So, am I allowed to start now?
Yes, and
Okay.
yeah, thank you so much, Roseanne. And to be honest, I personally, this is my first time meeting you, and don't know you too well, so I'm not sure if anyone in Phyllo could introduce you, or if you don't mind just introducing yourself. And
so I'll do that on the second slide. I will introduce myself because that's probably the best way to do it. So I was asked for this training today. As I pointed out to Laura, I've actually done this training in Anchorage, Alaska for the master gardeners there. I've also taught plant pathology 2 semesters there. Before that, I was a TA. And I literally took a course in plant pathology back around 1980. So it would be literally 40 years that I've been working with both plants and diseases of plants. So basic phytopathology will come before the first break. Types of diseases will come before the first break. And then we'll even get started on fungal diseases. And this is the outline that Brian made. But it turns out you would think that with 4 different types of diseases, that it's 25% each. That's not true. Fungal diseases are about 85% of plant diseases. So it's okay that we're spending a lot of time on fungal diseases because bacterial, nematode, and viral will have less slides. Where they occur, though, they're very important. So it's good to talk about them. And then disease management. If you know what the disease is, you want to talk about what you can do to minimize it. So that part is coming up. This is the title slide that Brian Bush, I want to give him credit because he was the diagnostician for, I think, over 30 years here. And he just retired in December. And we were talking outside, it was probably late November, when he said, he didn't really want to retire if he was leaving the position vacant because who would fill that need? And I said, well, I'm working part-time for the coffee berry borer project, and I have a background in plant pathology, and I have done informal diagnoses, probably most of that 40 years that I've worked. If somebody would bring something in the previous place I worked, it became like a puzzle to figure out what was doing it. So while I have experience, most of it was in Alaska. That was 30 years of experience. When I retired, my daughter has been here about 11 years. And 3.5, 4 years ago, she said, why don't I come retire here? And I said, you mean I will never have to worry about the pipes freezing again? And so I decided to move. And it turns out I knew that I could spend the rest of my life trying to learn tropical plants. So tropical plant pathology is not my background. It is Brian's, and I'm learning as fast as I can. The other part, that Brian worked 40 hours a week. I am only working 10 hours a week as diagnostician. So what I often say is I'm on the steep part of the learning curve. There's a lot of things that so if I hear it once, I can mention it again. But if I've never heard it before, I have to look it up on the internet. And Brian is kind and gracious. And when I ask him questions, he gives me a lot of advice and steers me in the right direction. So going back to the title, in Agricultural Diagnostic Service Center, ADSC. And this is found in many states. They had a big one in New York State where I went to school. We did not have any in Alaska because it couldn't pay for itself. And right now, Darren Park, who's in charge of the ADSC, he's working to make sure that it is a reasonable return on investment for a cooperative extension. So I'll get into that more. At this point, I wanted to make sure that it's not just me talk and you listen. So if all the people who are on, if they wouldn't mind typing into the chat one plant disease that they've heard of. And then I'm going to ask the agents to tell me some of those in a couple slides. So please, if you're on and thinking, just one that you've heard of. So Darren Park, mentioned down here, he asked me to reiterate this and point out that we have a permit to ship samples from extension offices to the Diagnostic Service Center on Oahu. Nobody should be putting a sample in the mail by themselves and mailing it to an extension office or directly to them. The permit is only from office to office. And I wanted to emphasize this because that's part of the importance of being on an island. If somebody mails a sample and they don't know what it is, what if they're mailing Rapidohea death and the package gets smashed? What if they're mailing coffee leaf rust or something else? That it is correctly under quarantine to mail plant parts, soil, plant pests such as microorganisms, insects, nematodes, and algae. And we have specific permission to do this. I have to include this permit on the outside of the box every week when I mail it on Tuesday. And I have to include some other things and keep a record of how much samples and what they were for. So this is just to emphasize that being careful starts with you telling people you can turn in a sample to an extension office, but you should never mail it directly. And now the next couple slides are, I wanted to put in pictures, but I also didn't want to overuse the internet. So if I had known how to minimize pictures, I would have had something more exciting than just 5 words. But as you know, we're on an island. That is why there's a permit. That is why when you get on a plane and come back here and miss your COVID test and can't come into the office, but you have to fill out, do you have any of these plants soil, or anything else that might not be in the island already? Are you importing any pests? So the first line of defense is to exclude pests and organisms and even plants that haven't been here before. In this day and age with airline travel, it's easier to carry seeds around to places that you don't expect. It's even easier to carry around spores and small insects. And if you have coffee beans or oranges or other things like that, you don't know what hitchhikers are on there. So usually excluding something is called a quarantine. And what I was taught in school that I want you to hear and think about is a quarantine buys time. It is not a permanent solution. You cannot stop the tide forever, but if you slow it down, you can plan what your response will be when it does come. The first response would be to identify it. Now it's kind of hard if you're familiar with what's here, how would you be familiar with something that's not here? You know, there are pest bulletins that are put out by the Hawaii Department of Agriculture saying, look for these, we don't want them here. The two-line spittlebug being a case in point, that it is wreaking havoc on pasture on Big Island. And, you know, it wasn't here a few years ago. So, you know, it didn't seem like one small insect could have that big a devastating effect. So when it comes to identify it as quickly as you can, and then you move on to manage it. And I would ask a question, but I can't hear your answers. What's the difference between manage and control? Well, it's literally a matter of semantics. They're usually manage is going to include more things, like you can think of cultural control and physical control. And in general, if you say control, people start thinking of chemicals, pesticides. And pesticide control is usually the last line of defense. You know, exclude first, manage. It is one of the tools in the toolbox, but you don't want to use it first. So managing is thinking of how many different ways you can influence the disease or the pest and minimize it. And if necessary, chemical controls are a very helpful tool. And there are controls that are biological control agents or coppers that are bactericides that are labeled for use in organic systems. So even just because you're spraying something doesn't mean that it doesn't qualify as organic. So that part about considering why you would be using the spray and does that affect whether you can say your product, it meets different standards. Usually people grow organically because you can get a higher price on the economic forefront, but there's also a lot of health benefits to doing that. So you want to think about your management and control to go along with the product at the end. All right, I think we have, I will show you guys, we have, after this, talking about island, change, education, science, diagnosis, and then we get back to pictures. Just so you know, it's coming. I wanted to say this because I think you already know this, that change is the only constant, was said by some famous scientist, I don't remember. You know, it's actually kind of cyclical or a cycle or a circle, if you wish, to the quote the Lion King, the circle of life. Things grow, bloom, fruit, and you either harvest them or they're harvested by fungi first. So decay, in my books, is being harvested by a fungus. That's probably a more ecological way to look at it. And, you know, the circle of life, if you didn't have decomposers, what would you have out there? You know, a pile of leaves and dead carcasses and other things. So in the tropics, things happen to decay very fast. So that part where the plant disease can get the upper hand And this becomes important because if you have a cause of disease, secondary things will move in and start decomposing, decaying the plant tissue. So sometimes if there was a particular sample that I got and they sent it on a Thursday and Friday was a holiday, so I got it on a Monday. And I thought that would allow it some decomposition and I could find more of the organism. But what happened is it was so rotten, I couldn't tell what came in first. And I had to say, please give me some more plant tissue because I cannot identify what the secondary things were as to compare it to what originally caused the decay. So I'm usually looking for the primary pathogen, but there's a lot of other secondary things that jump in and it becomes hard to say what was the original culprit. Okay, change is a constant. This is something that I heard in Alaska from a superintendent of schools. And she was saying that shift happens. And what she did is pointing out that in the old days, like when I went to school, I had to look things up in books. So a library was an actual treasure. Now you can find more on your phone than I could find in a library when I started school. So at the beginning, education for many, many years was trying to find information. You know, what is this plant? What is this fungus? Just basic, what is it? What is a name for it? Now there is so much information at our fingertips. You can talk to Siri or Alexa or use Google and you can find information really fast. So now instead of finding that information, you have to sift that information for relevance. So it's the information that you're looking at. Is it relevant to the question that you have? And I would pose, what is the difference between knowledge and wisdom? And it's somewhat of a rhetorical question, but knowledge is closely linked to information. You know, it's a set of facts that many people agree upon, but what do you do with it? So wisdom is knowing how to apply that knowledge. And usually when people are coming to you in a master gardener clinic, yes, they're asking for information, but they actually want to have relevant information, which literally becomes the wisdom of your answers. So the last bit here that I wanted to mention, because you should keep it in front of you, the source credibility when you search on Google. If I want to say that Wikipedia is a favorite place to start, but I would never cite Wikipedia as a source. So Wikipedia has good images, it has information that is easy to find, but anybody is allowed to add information and they may or may not have it backed up by scientific study. So usually by the time I get to a source, I might look at Wikipedia, but then I start looking for something from Cooperative Extension. California and Florida, as well as our own University of Hawaii, have a lot of sources on plant diseases. And so usually when you give sources to the public that's asking questions, you want to have it have a .edu, have it from an educational institution. There are some .coms, which might be from companies that are selling pesticides. Now they have credible information too, but they're probably trying to sell you a product. So if there's a choice between using this product or not using it, they're probably going to err on the side of use this just in case, or use this because it worked in California. You know, if it's not labeled for use in Hawaii, it's illegal to use it. So it doesn't matter if they recommend it. So that part about they have credible information, but it doesn't apply to Hawaii. So this part in education, It used to be information for the longest time, and now you have so much information. It's like a waterfall going into a teacup. You have to strain out the information that's going to be helpful in this situation. Is that too much? So this few less science questions. Science actually is questions. You know, why is that? Where is it? How much is it? All of these are questions that you can objectively answer. And then if you do that, you usually publish it in a paper and your peers critique you for it. And it's not very pleasant sometimes. But besides that, yes, I have written some scientific papers, and going through the review process, you know, they have questions like, why did you say that? Or what data supports that? All of these, science is not having all the answers, it's having good questions. And sometimes when I laugh, you know, and make sure that people understand that I don't know all the information, but I say, but I can ask questions with the best of them. And so when you're listening to stuff like this, I would rather have you ask questions than think that the first answer you come up with is enough and you don't have to keep looking. Even papers that I've written, I still go back and review, is there new data that means that it should be updated. You know, science is never resting on its laurels. It's questioning and questioning and questioning. And luckily, the experience is cumulative. So they say, I'm literally standing on the shoulders of giants. All the people that have written about plant pathology have enabled me to just read their findings and give information to you without having personally worked with some of these fungi, bacteria, and plants. So when I came here and started working with coffee berry borer, you know, I have only actually done a coffee trail run in Kau. I haven't worked in a coffee field, but I have read a lot about it. And this past spring, when we talked about coffee berry borer and coffee leaf rust, we had a person from Colombia tell us that they've had coffee leaf rust for a long time. And yes, on occasions, it's still a problem, but they have more than one way to make it a small problem. So this experience that they had in Colombia, they said, I'm going to tell you what works in Colombia, but in Colombia, they found out that what works in Brazil didn't work well in Colombia. What works in Mexico didn't always work well in Colombia. So just because it works in Colombia doesn't mean it's going to work in Kona. So coffee leaf rust, learning from other people's experience means saying what is the same and what is different from the situation that they reported about. This I will probably come back to a little later, but if you're talking about information, you have to say what it is and give it a name. And how do you classify organisms, both large and small? Literally in my lifetime, I went through places where they said animal, mineral, or vegetable, which said, you know, it's either not alive, mineral, or it's an animal, or it's a vegetable plant. Well, when they started looking under a microscope, they found out that there are things that they couldn't tell if it's an animal or a plant, and they had to expand these groups. We can talk about that more later, only if there's time. But going back to the small, the species names for fungi, the species names for bacteria, the species names for plants, they are changing because people get more information from doing molecular studies. And then they say, you know, this is a better fit for how to classify this species. So you may talk about a particular fungus, but know that there's a different name for that fungus possibly 20 years ago. And if they come out with a new name, then you want to look under both the old and the new to find information about it. There was a case where they were talking about coffee leaf rust Now I'm doing my best and I may not remember it correctly, but they said, no, it was a different fungus in Puerto Rico. And they said it had been reported in Puerto Rico, but it was probably incorrect. So they had a report of this, but it probably, that fungus actually became a problem later. And the first report was somebody's best guess that doesn't sound like it was right. So again, the source credibility. I give the naturalists that came here a lot of credit for being the first ones, like Darwin and the Galapagos. What do you start saying about these tortoises and finches that are seen nowhere else on Earth? You know, the Galapagos actually has a lot more relevance to us here because we have a lot of endemic species and we don't want to lose them. And in some cases, we haven't even identified all the ones that are here before they're starting to compete with other things that were introduced. So going back to the top here, science is about questions. You know, the answers are never certain. You can question the answers all the time, but asking more questions and looking carefully, that is good science. One of the times when I was in grad school, I said, you know, I do these experiments and the results always surprise me. And the professor said, that's good. That's what's supposed to happen. So it doesn't work like I predicted, but I learn from the results that I have. A quote that I've used many times is, in science, the next best thing to being right is being wrong. Because either way, you learn something. So please keep asking questions, which will help you do a good diagnosis. Now, this comes from years of experience working with cooperative extension, more in Alaska than here. But that part, if I see something and I say, oh, it's this, there you go. Like they won't even believe me because I didn't take enough time to listen. So the first thing you should do on Master Gardener helplines and other things for diagnosis, listen to what they're saying. You know, they might have some clues that you don't get the first time. When you're listening, you start to ask questions. Was it in the shade? Was it in the sun? Did you see it last year? Have you ever grown this crop before? Do your neighbors have it? So listen, start asking questions, and this is teamwork. Both the person, the client that's talking to you and yourself are both working to come up with an educated guess. An educated guess is often called a hypothesis. If you're doing an experiment, you guess what the results will be. And as I said, I have often guessed wrong and the results were different. So this first educated guess is good, but you have to repeat and learn from it. There's lifelong learning, that part of listening to what they're telling you, asking questions, gathering information that's relevant. And this cycle gets repeated till you think you have a good explanation and then you move on to the next thing. So even for the samples that I get in the lab, sometimes I have a quick answer, but most of the time I have to take time and look harder. And there are some times where I get a sample that I can't give an answer. I have to say that, you know, I couldn't, inconclusive. I don't like to say that, but that is an honest answer. And if it's important, I can ask for more samples. Now this part here, garden and paradise, I taught a course once called Leaves in Our Lives, and it was talking about how human civilization has used plants over the years in many, many ways. And it said that the root word for garden is also the root word for paradise. You know, there's historical context of the Garden of Eden was paradise. But usually when people are planting a garden, they are making something that is what they think of as a paradise. If you go over to England, you have these very neat and tidy gardens on big estates where they have lots of gardeners in time, because they were literally carving out something out of the wilderness. Now, if you look at my version of paradise, my garden, I call it naturalized, but I actually let things grow and just trim them as little as possible. So it's almost the opposite of an English garden, because I don't see enough wild in my life. I have lived in subdivisions most of the time, and I want to go someplace where it is less rigid. This is just me personally, but I don't have a lawn, and my goal in life is not to have a lawn. So the place that I purchased in Pune is bulldozed lava, and it would be very hard to put a lawn in, and I'm so happy with that. So when you're talking to somebody about a diagnosis, you also want to ask, what are they looking for? And be aware that it's important to them. Because if you're looking for your own little piece of paradise in your garden, then you're looking for a place of calm and happiness, and it's very upsetting when that balance isn't there. The last bit here is what I would hope is common sense. But if you work in the medical field, if you talk to a patient, what you talk to them about, it's confidential. You don't spread that. I have worked in the university. I've also worked in K-12, and specific students, specific incidents that happen, that's confidential. So I can say, I knew a student one time and this happened on the playground. But if there's any possible way, I don't use the names, I don't use the times and places that would allow someone else to identify who I was talking about, because that's a break in trust. This, going back to the part, if you listen long enough, you build a relationship that is trust. And if there's time at the end, I'm talking a lot, we could hear that Steve Beer story. So if you want to, you can ask me about that at the very end. And so this is back to Brian's picture. And when he was in the role of diagnostician, working 40 hours, he did both insects, so citrus leaf miner, nettle caterpillar, and I am only working 10 hours a week. I'm also working 20 hours a week with the coffee berry borer. So I don't do nematodes like Brian did. I don't do insects like Brian did. And it's not because I couldn't, it's because there isn't enough time in a day. And so I'm hopeful that if the budget allows, at some point they will hire another diagnostician. And I hope they hire that person to work in Phyllo, because Phyllo has more agriculture and a larger landmass. So it is a good location to ask, what is this? What disease? What insect? Living here in Pune, we're the center of little fire ant, of coqui frog, of, I have been here for three and a half years, and I've just watched the ohia trees die in the yard. One branch will wilt and leave the leaves on. Phyllo and Pune, with all of the rainfall and all of the agriculture, that it is a place where more of this happens. So I was wondering if I could hear what some of the diseases that were written down by people. So if some agents would read at least 2 diseases from.
We had powdery mildew on.
Powdery mildew, yep.
In the chat, I see root rot.
Oh yes, we have a lot of root rots by different things.
We also have Huang Long Bing, HLB.
So I'm not familiar with that one.
Oh, that's another name for the citrus greening.
Okay. sharpshooter.
Citrus rust, which I've not familiar with. Black spots on papaya.
Oh yes, papaya tastes really good, and there's a lot of things that bother it.
And someone just typed in Rhizoctonia.
Now, that's a person after my own heart, because when I was in Alaska, I worked on Rhizoctonia for 10 years. And I'm intimately familiar with it. If I had stayed as a technician, I would have just spent 6 hours a day on a microscope. And that's actually part of the reason I went to grad school, is like, do I want to stay on a microscope this long, or go learn about a more diversified set of things? So Rhizoctonia is a worldwide problem, and there's different strains of it. Fusarium is another name of a fungus that it has what's called forma specialis. So it's like cultivars of citrus. You know, they're similar in one way, but they taste different. So Fusarium forma specialis, one of them affects one crop more than another. And it's actually very hard to tell them apart. The molecular ways have been a big help. Any other comments for common diseases? I think we got 2 from 2 different people.
We had anthracnose of mango.
Anthracnose, yes. It's usually a superficial problem that the same fungus, if it causes something inside the coffee berry, they had to give it another name beside anthracnose.
And then
So
the
they
other one
called
we,
it coffee berry rot. Yes.
the other one we have is bunchy top over here in Phyllo.
Yes, banana bunchy top is a virus. And the sad part about that is, if you have it, you're supposed to chop everything down and tarp it so that it can't be spread by aphids to the neighboring bananas. So for a small diagnosis, it's a huge step. I worked at a school. The rainbow in this picture is from a school in Pahoa, and I helped with their community garden at the school. And one of the things they were doing was chopping down the whole banana patch because of bunchy top. It's very sad to watch school children chop down bananas. Okay. Some of the other slides will go faster than I have been, because I realize in 9 minutes we will have a break. This is the diagnostic or analytic services provided at Manoa. And these first analyses, so analysis with an I means there's one type. Analyses means there's more than one type, because irrigation water and nutrient solutions. Most of these analyses are going to be used more by commercial growers than homeowners. So it's good. They bring in a sample, they measure a certain weight, they put it through a certain machine, and they get a number. And that number goes usually to the extension agent, and the extension agent makes recommendations or not, depending if they have something published data to recommend it from. But you can predict how long these analyses will take. Now, these last 2 down here are identification. If you have enough of the insect, it's fairly easy. Well, it's possible to identify adult insects more easily than the larva or immature forms. But still, if you have enough specimens of an insect, it's microscope work, usually. Now, with a plant disease identification, when I was talking to Darren about this, I can't predict how long it will take me to get an answer. Sometimes it will take me 2 hours, but if it's not clear, then sometimes I have to wait and let it grow on the plant tissue. Sometimes I take a small piece and put it on agar, and there's different types of agar, and the agar has to be made in advance. So there are probably at least a third of the diagnoses that I've done have taken 2 to 3 weeks. So these 2 identifications are less predictable as to how much time it will take. And it's harder to say which is a good investment of time. Now this part Brian likes a lot. ADSC also sells University of Hawaii developed fruit and vegetable seeds. And this afternoon we'll be talking about rainbow papaya seed. Rainbow papaya seed was developed by a plant pathologist who I met, Dennis Gonzalez. And it was developed to be resistant to papaya ring spot virus. So there are times where this is an example of a GMO rainbow papaya that was around before people had more genetically modified plants and they became more of a concern. So there are some times which says any GMOs except for rainbow papaya, and sometimes not. But that part about selling fruits and vegetable seeds, please be nice to Darren about that, because it takes a lot of time and effort to count them or sort them or clean them. All right, just how much is this going to cost me? This is very true, nominal, because the prices for insect identification are $7. The fees are not likely to change. The fee for a plant disease diagnosis is $12. And sometimes just the cost of materials is more than $12. But that part where for many growers and home gardeners, $12 is a really good investment. The fees are collected before test initiation. There was one particular case, there was a person that came to Phyllo and he brought a tea leaf plant with some yellow leaf spots. And I said, oh, that's Cercospora. And, like, what do you want to do about it? Fertilize the plant. So Darren had said that if I don't look at it under a microscope, I don't have to charge for it. Advice is free. And a lot of times in Phyllo now, they actually say, if you're a home gardener, you may want to wait and ask the home gardeners on Friday, because that's free. But that's also because I'm one person and I only work 10 hours. And it's really good if you go to the master gardeners first. So that part where even if I can look at it and give a reasonable explanation, you don't have to make it a sample. But if it's important, then you do that. Then I spend time under the microscope or materials in the lab, and it goes from there. So this is quickly taking a soil sample. Does my soil have problems? Does my crop need fertilizer? How much? One of the things down at the bottom here is if it's representative, if the sample does not truly represent the soil you intend to treat, then all the precision of the analytical process is useless. So they said, how many soil samples for what size field? And the bottom line is, some of my professors, they said, well, it's one soil sample for an area that you would treat differently. So if you have a 2-acre field and you're going to fertilize it all the same way, then one soil sample will be enough. If in this 2-acre field is divided into half-acre plots, that would be 4 different plots. If you're going to treat those plots differently, then a soil sample for each plot would be a good idea. So it's not a matter of how much space, it's what is going to be treated differently. Please make it representative of the area that you're going to treat differently. And these are good things to look up. There's also a cooperative extension service about how to take a good sample. So I'm going to go through this kind of fast. The accuracy is as good as the sample provided. Sometime if I'm testing for a virus, I can say that it was not detected in the tissue that I tested. But if there was a different part of the plant that was showing more symptoms, it could be that the plant has the virus and I just didn't find it in the piece that I had. So containing the right material, again, fresh and in good condition. I mentioned that sitting in the UPS over the weekend, a long weekend, meant that I couldn't do anything with it. And then the source data, you know, when and where. We usually ask for the altitude, you know, it has to do with temperature and salt spray and stuff like that. The more information that's submitted with a sample, the easier it is to narrow down the diagnosis. So a good thing would be to have some of the healthy plant versus sick plant, so I know what each looks like. The symptom description, it's like, are there patterns? The easiest one would be, did it just die overnight, or has this been going on for a couple months? Are the leaves slowly turning yellow, or did the whole plant wilt? The distribution and spread, is it on 5 plants or 150 plants? And how fast did it spread if there's 150 plants? So the history, did you plant this crop the year before? Did you have a new seed source? Was the weather different? The growing conditions. My example for growing conditions is here in Pahoa on Big Island in the south. They had a hapu'u fern planted in the center of the traffic circle. And while I loved seeing it there, it was sitting by itself in bright sunlight and it was not doing well. And then somebody ran over the middle of the traffic circle and they replaced it with something else, because as pretty as a hapu'u fern is, it was out of place in the sun and low humidity. Cultural practices. Was it fertilized? Was it irrigated? Was it sprayed? That can make a difference in saying what the problem might be. So for your diagnosis procedures, you probably want to review the cultural practices that they have done. Was it planted at the right time in the right depth? Have you been fertilizing it? Did it need any pesticides? How often do you irrigate? In my experience, when I had houseplants in Alaska, I never used liquid fertilizer because I would usually wait till my houseplants were very dry and then I would get fertilizer burn. So I would use Osmocote because the irrigation frequency for houseplants was more important to avoid root rot, and therefore it was a high likelihood of fertilizer burn. So reviewing cultural practices, environmental conditions. You know, we've had a lot of rain. Is there a drought? The soil type. You know, here I'm where there's a lot of lava and it drains really well. But in a lot of other places, if there's standing water, there are many plant diseases that can move through that. And this last part, check host specificity. So if it's on a bunch of different plants, if there's a corner of the garden and all of the plants in that corner have yellow leaves, is that because they don't have enough nitrogen, or was the nitrogen leached out because there's a lot of water in that corner? So many hosts indicate an abiotic problem. So biotic means life, abiotic means not living. So too much water would be an example of an abiotic problem. Too little water is also an abiotic problem. And we are now 2 minutes past time for a break. So I'm going to leave it on this slide here, and I'll see you at 2 minutes after 10. Is that
Thanks,
a good plan?
Roseanne. Sounds good.
Okay.
Thanks, Roseanne.
So I imagine that most people are back by now.
I think most of us are.
Yep, we're here in Hilo.
Very good. I just love flipping through these slides. This goes back to the beginning, saying what we were hoping to cover. And pretty much we will be covering types of disease and fungal, bacterial, and nematode and viral diseases in this next hour. And then there'll be another break, and then we'll have a little bit on disease management and questions. And I was very happy to see all the comments in the chat box. Was there like 60 of them? I would actually ask if you would type in one gardening plant that you like to grow at this point. So in 5 or 10 minutes, I will ask the agents to tell me what those might be. But this was the last slide that we stopped on, and we're pretty close to on time for where I was expecting to be. What I wanted to say, we've covered it mostly, is that diagnosis, it's as much an art as a science. Certainly the information is based on science, but asking the right questions and looking in the right places, knowing when to ask for another sample or what questions to ask about cultural or environmental conditions, it is an art as well. So this was Brian's slide, and we can go from this, a very good sample, because you can see healthy plant tissue and you can see places where it's diseased. It's brown, there's something wrong, versus something like this. And in the lower right here, there's a plastic bag with some potting mix and possibly a decomposed piece of something. It would be very hard to tell anything from this. Although if this was a sample for nematodes, that's possibly useful. So just going over the comparison between a good sample with healthy and diseased, this sample here, basically I would say, please give me some fresh tissue, because what's left here is not representative of probably what was out in the field. Here's another good sample because it's a dry paper towel. So the plant itself will give off a little bit of moisture. The paper towel will help keep it there and actually wick some of that moisture up. If you put it with a moist paper towel in a plastic bag, again, everything that's likely around to decay the stuff will go to work, and this is hard to tell what is secondary and what was the primary pathogen. This is another example of a very good sample because the root system is packaged in its own bag that was kind of held with a twist tie or something. So you can see what's going on with the top of the plants, and you can also look down at the roots. The root rot that they were mentioning, there's some things like Pythium and Fusarium and Rhizoctonia that are on the roots. So you can see problems with the leaves up here, but I could never find Pythium from a leaf. I would have to look on the soil. Now this works well with small potted plants, but what if it's a tree or a shrub that's outside? What I have found sometimes is if they can bring in pictures of the tree as well as some of the leaves, it's hard to do much with the roots of a tree because you don't want to hurt it. But that part, it's like, what can you do to give the biggest picture to the person asked to make a diagnosis? And then this is turf correctly packaged, that it's dry newspaper, so it's wicking up the extra moisture. And in the top right here, we have healthy plant tissue. It looks like a mum to me. Over here, you see some plants that are not looking so well. And I would first off, do you think this is bothering the leaves or the roots? What do you think, Kalani?
Sorry, what was the question again?
Do you think in this case that the pathogen is bothering the leaves or the roots more?
I don't think I can see clearly enough, but I would guess for the leaves first.
Okay. Well, that I would say make another guess. Guess again.
Well, I guess if it's uniformly dried out and it's something that maybe happened pretty quickly, it could be a root issue.
Yes. So both, as I said, the next best thing to being right is being wrong. And I don't know that this isn't a leaf problem, but when it affects the whole thing like this, it makes me wonder if the roots are not functioning well. So the upper plant is suffering because the roots aren't there. Nematodes do this a lot of the time, but that part, because you have both what the healthy plant and the disease plant, you can compare the two of them. Whereas here, you don't even know what healthy looked like. So the part that as master gardeners, as a diagnostician, I can always say, can you please bring some more if it's necessary? I can also say, here's a handout from the extension saying how to take a good sample. And they have these, the same for Kona coffee nematodes. There's literally a handout for how to take a good sample. So there's a lot of different ways to get a better sample. Now this is back to the beginning, just saying a review, and I don't need to go there again. Plant pathology, the diseases. This is going to come up with a big mouthful right here. And then the disease triangle is more fun. So I'm going to do both. I'm going to read that in just a minute, but I'd like you to look over here because that's more colorful and actually more important. So reading this once, phytopathology. So this is plants and pathology is diseases. So phytopathology is literally the scientific way of saying plant diseases. It's defined as the study of the organisms and environmental conditions that cause disease in plants, the mechanisms by which this occurs, the interactions between these causal agents and the plant, such as the effects on plant growth, yield, and quality, and the methods of managing or controlling the plant disease. So it's kind of a whodunit. You know, what organism is affecting what, and what conditions cause that, and how do you stop it? So coming over here to the disease triangle, this red center is what we want to avoid. Usually we want to grow plants that have no disease, but there are 3 different parts that all have to be in place before you have this disease: a susceptible host, a conducive environment, and a pathogen. So if we use coffee leaf rust as an example, it's a pathogen that has been in Hawaii for less than a year as we know it. So it could have been here before that, but it wasn't identified till last year around Halloween at the end of October. And so before that time, we had a susceptible host of coffee Arabica plants. We have a conducive environment because there's lots of rain and high humidity, but we had no coffee leaf rust because there was no pathogen around. That was a lovely place to be in. We excluded it for as long as we could. It actually turned up in other coffee growing areas back before 1900. And then in the 1970s and 80s is when it became a problem in Central America and South America. So this pathogen has not been at a worldwide distribution until recently. So the disease wasn't a problem. What happened in Central and South America is they started planting resistant host plants. And here in Hawaii, we have already started testing resistant host plants before the disease came here. They're in quarantine in Oahu, and they've been testing them to see how well they grow here. But in Colombia, as an example, they have resistant host coffee plants. So without a susceptible host, you can have coffee leaf rust and a humid environment, but there's no disease because the host is resistant. So this is a good way where you don't have to spray or do anything else, but it means replanting all your coffee trees. So that's a long term if you want to replant some resistant citrus. It takes a while for it to grow and produce fruit. There are things like basil. I happen to grow Thai basil because basil seems to get every plant disease known to man. And it's very susceptible to a lot of things, but there's a few types that are more resistant, and that's what I chose to grow so that I don't lose them as fast. The other part that management is often changing the environment. Like you can cut down the weeds with better air circulation, and they have found out for some plant diseases that that's just as good as spraying because you are making the environment less humid, there's more airflow, the spores get blown away rather than landing in a moist place. So this disease triangle is more important than this big, long definition. What you're trying to do is eliminate any one or two of these so you won't have disease. Now, abiotic, I believe there's only 2 or 3 slides on this. And as I said before, abiotic means non-living. So it's also non-infectious. And I will go down this list and talk about them a little bit. I don't exactly know which these examples are because Brian put them in, but you can tell that on this side, these plants are not doing well compared to that side. These trees, something happened that they are not providing the landscape cover that was probably expected. Now that's easily too much or too little water. And here in Pune, we know about both of those. Nutrient deficiencies or toxicities. So if there's a lack of nitrogen, phosphorus, potassium, but then there's some microelements that sometimes become deficient. Iron deficiency will give you a very yellow plant, and it's actually hard to make that plant recover. Toxicities can be when there's too much of something. So if you have some micronutrients and you want to add, if a little is good, more is better, you can ruin the soil by adding too much. Because if you don't have enough, you can add more. But if you have too much, you can't take it out of the soil. Along with toxicities, there is a spray for coffee leaf rust that involves copper. So how many times can you spray copper before there's too much copper in the soil? No, there's a lot of factors in considering control. Too high or too low temperatures. We have lovely modest temperatures here. It's sometimes too high. It's not very often too low. But where I come from in Alaska, frost damage would be an abiotic disease type. If you go on the East Coast in vineyards, they usually plant vineyards on a slope because there's airflow going down the hill, and that means it's less likely to have frost. But apple trees and grapevines, if there's a frost in the early spring, it kills the blossoms. Therefore, the blossoms don't develop into fruit. And it was all because of low temperatures in that spot. Lack of oxygen. The best I could think of is if you have water on the roots, then you have water, but you don't have oxygen and the roots die. I have seen a tree on a lake and the lake level rose 18 inches. And this big, beautiful tree died because the tree's roots didn't have oxygen for 6 weeks. And then the lake level went back down, but it was too late for that tree because it needed those roots to be functioning. Pesticide toxicities. It's also called carryover. And it's the reason that pesticide regulations are very carefully made and should be strictly enforced. So the label is the law, please follow it. So this does not happen in a place near you. Air pollution. We don't have as much of that on most islands, although I would say here in Pune, we have seen it because of the volcano. When the East Rift Zone three years ago, it was here for the whole summer. And I happen to be upwind of it, which I'm very fortunate. But if I go down to some of the places now, you can see which trees were more sensitive and they died. And then a few of the usually invasive species that could go with the air pollution. Too much or too little light. You know, anthuriums look terrible if they get too much light. There's a lot of other things that if they get too little, they don't produce. I was told that here in Puna, we get enough rain and cloudiness that there might be too little light to get a good tomato crop because there's so much rain most of the time. So that's something that you can use shade cloth, and then you can cut down. Coffee in shade is usually where coffee leaf rust will show up first. So you might start growing your coffee in more sunlight if that's your goal. And then cultural practices, weeding, hilling, stuff like that. I think we have a few planting mistakes. Can somebody tell me what the problem is here? In the lower right, all this brown stuff is probably a flood. So what's the problem? This abiotic disease that will stunt these plants.
They'll just be drowning, lack of oxygen.
Yep, exactly. Now, going above that, this planting mistake, I actually don't know for sure what caused these tips to die back. This one, I do know. This one, I do know. I'll go with on the left first. It looks like the soil level was here, and where the plant wanted the soil level to be about here. Now, in Hawaii, there are some plants that don't care much. If you put cut off a tea plant and stick it in the soil, it will root along the stem. But there are plants like strawberries being an example that they usually give, planting it too deep or too shallow. And this tree did not do well by being planted too deep, or shrub. Now this one in the middle, you can almost see the shape of the pot, can't you? And I actually bought a couple fragrant tea plants that were pot bound like this, and they sold them to me at a quarter of the usual price. But about 4 months later, the plants, even though I transplanted some of them, they all ended up turning yellow, and you know, even that deep discount, it was just a couple months of pretty plant. The plant could not overcome these circular roots. Sometimes you can try trimming them and putting them in, but in general, if a plant is this pot bound, it's not going to recover easily. And that's not because of a disease, it's just because of a cultural practice, that it was allowed to get too big in a small pot. Okay, planting too deep. Another example, we'll go through it fast. If this is the recommended depth and it was actually planted a little bit deeper, but you put mulch around it, there was a lot of organisms that started rotting the bark here. That's usually not enough roots to support a plant like this. So planting too deep can be an abiotic disease. This, just quickly, volcanic pollution. Here in Puna, we actually saw quite a bit of this 3 years ago. And then we had 2.5 years with no sulfur dioxide being emitted, and Kona had the most beautiful sunsets. And still there's a lot less, but if you take sulfur dioxide and mix it with rain, you get sulfuric acid. So you're literally burning these leaves, and a lot of this metal will rust. Like I said, I was upwind, so I didn't get this in my yard. But if you're getting this, there's usually not too much you can do other than relocate or choose different varieties. You know, we had 3 months of it. People had to leave their farms. Some farms were covered, and you know, quite a few things have recovered fast. Some of them, this one might recover, this one, you know, but this is unique to a certain part of Hawaii. And I have personally experienced it, and I'm glad that most of you probably have not. Okay, this is the fun part. So now we go to living organisms, biotic or infectious disease types. And again, we're talking mostly about fungi. There'll be another slide about that. But there's also viruses and viroids, bacteria and phytoplasms, also called mycoplasms, nematodes is up here, higher plants, mistletoe, dodder, witchweed. There's a very nice example of dodder on the way to the Hilo airport, and only a plant pathologist would think it's a nice example, but it is a plant that looks like it comes from outer space. It's these orange strings covering a tree. And I've seen it on the East Coast and I've seen it here. Protozoa sometimes cause a problem, etc. Means that there's always an exception. So this picture over here is showing the size relation. And this outline here is a plant cell. Actually, if you look at African violet flowers or orchid flowers, you can actually see plant cell size with a hand lens. They're fairly big. This part that I have, whoops, there we go. That is an example of a protozoa we'll come to. But this would be the cellulose, and it's outside the cell membrane. If this is the head of a nematode, most nematodes do not have this stylet. But if there is this stylet, this cut, it's like a little needle to punch into the plant cell. And so nematodes that have a stylet can pierce the plant cell and then get the sugar and other nutrients out of it to the harm of the plant and the benefit of the nematode. Over here, you can see fungi. So you can see that these fungal cells are much smaller than the plant, and they're thread-like. Going on, this corner is a bacterium. This bacteria is smaller than the nucleus of the plant. So bacterium, very small, very abundant, and they're single-celled. Mycoplasms are one of those lovely related concepts. They don't have probably the same cell wall made of sugars that bacteria have. Going down to the very small things, there's viruses, different types, different shapes, and then viroids are even smaller than that. So tobacco mosaic virus, cucumber mosaic virus, you know, they have different shapes, but in this age of coronavirus, it's some nucleic acid surrounded by a protein coat. And just to say it once while we're here, they always ask, well, is a virus alive? Well, technically, it can't live by itself. It cannot reproduce by itself, but it is a separate entity from the host cell where it reproduces. So I always gave my answer is it's on the verge of life. You know, it does some things that living things can do, but it can't actually exist outside of, it can't reproduce outside a host cell. It can actually persist sometimes. But I like that this gives you the size differences. Now, because we looked at different things, does anyone have a guess for what this might be? I believe it's an avocado leaf. Picked from my neighbor's yard a couple months ago. There's a parasitic algae called Cephalurus, And there's two different species. One does top and bottom. It goes through the leaf. The other stays right on top of the leaf, but it makes these spots and that blocks out sunlight. And it also takes sugar from the plant. So it is slowing the plant down and it is very visible. Usually, you know, a little more sunlight and that the algae will not have the upper hand. But this is a case of it's not a fungus, bacteria, it's actual protozoa. So this is from Brian. Anthracnose on papaya, leaf spot on rose, Phylosticta on dendrobium flowers, powdery mildew was mentioned, root knot nematode. Even though this is an animal, it's still considered plant pathology because these are what healthy roots look like, and these diseased roots cannot take up water and nutrients as well. To clue you in, this is actually an example of an aerial nematode. It's a very uncommon thing, but it can be a problem and stunt the growth. This is a very common thing, botrytis. I was asked on an exam, like, what disease would you work on and how would you do that to write a grant? And I picked botrytis because it affects so many things, but it usually affects the delicate parts. So these strawberry fruit that you see here, like if you're thinking from the strawberry perspective, they have made their little seeds, and at this point, they're just looking for the seeds to be distributed. But we're looking to eat the fleshy part. So this is no longer appetizing or good for us. But for the plant, it's so close to achieving its purpose of making small seeds that it's not as big a problem for the plant as the rest of us. Now, when it goes back into the stem and kills the stem because it's in abundance, that is a problem. But that part where this is a banana leaf, obviously it's not doing well. So what do you tell the person? You know, if you cut it off and lay it on the ground, a lot of times what's on this leaf is going to have spores that go up. So it's usually remove the diseased plant parts. With coffee leaf rust, that's important that you spray before you remove them, because even moving them is going to release spores out onto the other coffee trees. So do I expect you to memorize all of these so that you'll know them off the tip of your thumb like I don't? I would expect you to do similar to what I do, is you look it up on the internet each time you work with something. So even this one that I've seen several times before, I look it up again. This one I see on a lot of different crops, but I look it up again. So don't try to memorize things in this day and age, but just know where to look. So fungal diseases, and as you'll find out shortly, this is 85%. We won't spend as much time on these. I wanted to let you see these words so that you know what they're talking about. Hypha is a single strand of a fungus, like we saw in that plant cell. So it's like a very tiny thread, smaller than a strand of hair. Mycelium is the name for many hypha. So this is a single one, this is plural, when they're in a mat or something that you can see with your naked eye. Sometimes if you break open a log in the woods, you can see a mat of white growth, and that's mycelium in its natural habitat. A spore can be single-celled or multicellular, but basically that's usually going to be something that propagates the fungus. It blows or splashes or gets moved. So this can either be a carbon copy, an exact copy like a strawberry runner or a sweet potato cutting, or it can be reproduced with recombination, and that would be a sexual spore like an ascospore or a basidiospore. So you'll hear these names, you'll see these names. Again, you don't have to remember, you can look them up each time. So Fungi are not plants, although I do have a book that says they are. It has changed that much in my lifetime. They are a filamentous organism growing as hyphae that are for the most part microscopic, and some produce structures such as mushrooms, seen here. 85% of the plant diseases are caused by fungi. So that other 15% still can cause damage, but if you learn a lot about fungi, you'll be covering most of it. And going back, are all of these plant diseases the majority of fungus? Absolutely not. If there are about 100,000 described species, most of them, like 92% of them, are beneficial or a saprophyte, which means it just decays things in the woods. Only about 8,000 fungal species are known to cause plant diseases. Although here on the island, the species that cause rapidohea death are new species. So we have not described all of the species that there are. And especially with molecular tools, we're finding more and giving them new names. But that part, when I traveled, which is the reason for my self-quarantine, I was reading the Hawaiian magazine, and it was talking about mushroom growing on the Big Island. So people, they have now listed mushrooms as a crop of importance and how many thousands of pounds are grown in a year. But in that article, they were also saying that fungal species are now classified as the largest organism on the planet. Because if you look at that mat of hyphae that together form mycelium and see if it's genetically the same, they say that there is acres covered by small threads in the soil that are genetically the same. So if you calculate what that is likely to weigh, that colony of fungus is probably the largest living thing on the planet. And when you have, whales here in Hawaii and redwood trees and other things that are very big, they started realizing that some of these, it's like having a huge strawberry plant that covers 2 acres. Or a creosote bush was one of the things that they said that it's all the same. So fungi are in places that we don't see, but they're doing a lot of things in the soil, and most of them are helpful. Only about 8% are causing diseases that we don't want. So this is a picture of a small bit of fungal growth called hyphae is more than one, hypha is a single one. So A and B are comparing, usually you talk about you have one cell and one nucleus. Well, this is a place where there is no dividing lines. Cenocytic is how I pronounce it. And bread mold or water mold, they move their nutrients so fast that they don't even put any cross walls to stop that transportation. Most other fungi have these cross walls, or called a septum, with a little pore in it. And here you see that there's neat 1 nuclei per cell. When I worked with Rhizoctonia, there was binucleate and multinucleate Rhizoctonia. So the binucleate ones would have 2 in each cell, and the multinucleate would have 3 to 9. So they're not following the rules that mammals and bigger plants, vertebrates, follow. So this is going on to plant pathology. It's showing a fungal hypha like powdery mildew, and it penetrates the plant cell wall. And then you have the membrane of the fungus directly next to the plasma membrane of the plant cell. So it's like if you take a balloon and you stick your fingers into something, you're not actually touching it. There's a membrane between the two of you, but by having all of this surface area, you can absorb more sugar and harm the plant. So one of the cases with fungal, they were trying to decide whether it was a plant or an animal. And they ended up making some more kingdoms. So instead of just plants and animals, then there came bacteria. And then there became plants, animals, bacteria, and protozoa. And they still couldn't decide where the fungi, if they should be a plant or a protozoa. They weren't a bacteria, they weren't an animal. But it turns out that they decided, based on how they eat, that fungi would be a fifth kingdom. And that's because how do plants get their food? They have chloroplasts that circle here, and I say they make their food. They use photosynthesis and carbon dioxide from the air to make sugars. They capture the energy. And animals usually ingest either plants or other animals. So ingesting food, it goes down one way. If I put food in my ear, nothing would happen. I have to put it in my mouth. It has to go through my digestive tract. So we get food and we absorb the nutrients in one place in our digestive tract. Well, fungi pretty much lay in their food. They secrete enzymes outside and they absorb stuff directly into there. They have, they absorb their food is how they decay other materials. One other thing that I might as well tell you, since you're all budding biologists and want to know, this plant cell wall is made of cellulose. And cellulose, that's what we make paper out of, everything else. And when they said that a fungus is like a small plant, well, the fungal cell wall is not cellulose, it is chitin, like an insect. So sometimes they have used chitosan. They put down things from crab shells, and if they start decomposing the chitosan, then they'll start decomposing the fungal cell walls too, and it helps with disease control. And that is true for most fungi, but there are water molds and their cell wall is made of cellulose. So then there's like, maybe it shouldn't be a fifth kingdom, maybe it should be a fifth and a sixth kingdom. And biologists, you have the splitters and the lumpers, and they have lots of work to do. I just knew you wanted to hear that. So this I'm showing you because I don't want you to remember it. I just want you to have seen it once. There are basically 4 different kinds of fungi that they usually talk about. When I talked about the water molds that they have cellulose in their cell walls. So Phytophthora and Pythium are 2 that you should hear about. We have Phytophthora here a lot. It usually spreads on the water. Phytophthora, phyto means plant, and Tophthora means destroyer. So when I talked earlier about late blight, which is the organism that caused the Irish potato famine, it was Phytophthora infestans, and it killed the potatoes and caused the famine. Pythium is another water mold. I have seen it once on cucumbers here. I have seen it in Alaska the same way. And if you control water from one plant to another, then Pythium can't spread. But if you have some surface where the water can go from one pot to another, then the swimming phase of Pythium or Phytophthora can swim in that from one plant to another. So for both of these, separating so that free water doesn't go from one plant to another is one way to help. And again, they're saying that it has no cross walls in the mycelium. So this is also sometimes called oomycetes. There's more than one name. This Deuteromycetes, imperfect fungi, I think this leaves a lot to be desired, because just because they aren't practicing sexual reproduction doesn't always mean imperfect in the whole world. But that is why they're called imperfect, because we don't know where the genetic recombination occurs. So that's what they say, imperfect stage. It is just like the strawberry runners or sweet potato cuttings, they are all genetically the same. They have cross walls in the threads, the hypha, and their microscopic reproductive structures are like this, looks like the head of penicillium. And these little spores break off and float on the wind. They land someplace and they start a new colony. So there's a lot of wind-blown small microscopic spores. Now, ascomycetes and basidiomycetes, they're both technically perfect. This asco means they have a little sac, and from that sac, they release 8 ascospores, which are not genetically the same as the parent. And in this case, most mushrooms, like we see here, they have little clubs that have 4 basidiospores hanging out on them. And again, they release them to the wind and they go, they're not genetically the same as the stem and the stalk. The little basidiospores drift and start new colonies. So just to let you know, because I'm a plant pathologist, these ones, the little cup fungi, are called discomycetes. And if you grow up in the 70s to have something called discomycetes, it makes you think of a little dancing fungus. Anyhow, I just thought you should know that. So here, again, I will say these words so you've heard them once, but even I don't need to know this because this is leaf spot on wheat. And we don't have a lot of wheat up here, but it is a nice picture showing you either genetically the same, how these go through the season and make secondary cycles, or this ascospores, these are not genetically the same and they are windblown. So this cycle is all from rain splashes and it's nearby. But when you have this overwinters and then blows further to different fields. And when you look at this, I want you to know that the Phylosticta on orchids, we have a fungus up here that is very common on orchids, and it does the same thing. There is Pycnidia, which are genetically the same. They produce these little spores that splash around a lot. And then they also produce Parathecia, which they come out in a little puff of wind and they blow over a couple, from one end of the greenhouse to another. So they spread it further away. And both of these types are part of the disease cycle. So I'm going to say this once, Septoria tritici, Microspirella graminicola. This is the way I pronounce it. I will let you in on a little secret. The best way to pronounce a scientific name, did you ever wonder? With confidence. You can say it however you like. If you say it with confidence, they're likely to believe you. But this is not the only way to say it. It's the way I've heard it said before. So when you have these little leaf spots here, this little leaf spot here will spread and spread and spread during the rains, during the season. But how does it go from season to season? The debris that's left in the field has both of these, and they wait till the next season. This will infect the seedlings that are in the same field because it doesn't move that far. It only goes on rain. And this can blow from one end of the field to the other or across the road to another field. So usually when you have a crop plant, you can look up on the internet and it will show you this cycle and you can guess how to break this cycle. So here you can bury this so it's under the ground and they don't have any place to fly and splash. So burying the debris further is one way to stop this leaf spot of wheat. Spraying in the middle of the season, if necessary, is one way to stop it. Just things like that. These disease cycles are not to be memorized. They're to be looked up when you need them. Okay, I wanted to show you this because this is not wheat. This is a banana, and it's caused by a fungus with the same genus, Mycorrhizal musicola is how I would say that. And again, I'll say it with confidence. Other people could say musicola, and I would still believe them. But this leaf streak, do you think these bananas are going to produce as much? Nope. So usually by the time it's at this point, I'm guessing, because I haven't looked this up, but if you cut off this leaf, you're going to want to tarp it or bury it or do something so that the little spores that are here don't fly over onto the healthy leaves. So that's true with a lot of plant diseases. If you have a diseased plant part, you don't want to leave it in the field for it to blow onto the new plants. And just again, you have these names change, but you can look them up on the internet and see the old and new name and go from there. So here, this was one of the favorites. I have seen this literally for 40 years. And this is under a microscope with 40x magnification. These are literally little grape-like structures of gray spores that blow on the wind. You know, a lot of times they don't blow that far, it's more touching. And so one of the ways for botrytis is usually not to spray for this, it is to improve air circulation. You know, get rid of the weeds, trim back some of the plants. If there's good air circulation, there's less likely that this will get started. But gray mold is very common, and it's not a very strong pathogen. So that part of the cultural control is usually to change the environmental conditions. But I have seen this on many types of plant species, and it's usually going to be the most delicate parts, so the flowers or the fruit. If it's here in abundance, it may start moving over to the leaves, but that's usually not where it gets its foothold. Okay, black spot of rose, Diplo Rosii. Now these leaves, this inset here, you can tell that they're not happy. If you looked at this under a microscope, the spores would be distinctive. And if you were going to spray for it, you would want to know that that's what it is before somebody chose to spray. But on these leaves, usually at a certain point, the plant will just drop them. But here you see these really sad flowers. My guess is that someplace along here, there's an infection on the stem. So if you have this same infection that's on a stem, it basically cuts off the nutrients to everything past that and causes wilt like this. So probably by the time it's in this phase, you know, you're going to prune first. And whatever you prune is going to be put someplace where the spores can't blow elsewhere. And then you'll decide if there's less than this, if you want to spray first, or if you want to grow a resistant variety. And that's one of the parts that you're so important, is the sooner the better. So I'm going to go to one more slide and say it is 10:51. And believe it or not, we are still close to on time, but I would rather have you guys get up, wake up, body break, and I will see you in 10 minutes.
Sounds good. Thanks, Roseanne.
You're welcome. if any agents are still listening, you could tell me some of the questions so that I can be thinking of them for the end. Only if you're available.
Hey, Roseanne. Not too many questions so far. There were a lot of responses to your, you asked everyone what they're growing, and there's
Yeah,
into the chat. I mean,
and
there's
that's
a lot of diverse.
That's wonderful. And that is what I'd like to start with.
Oh, great.
At,
Yeah, so pretty much
at
any
11.
and every vegetable and avocados. Somebody's growing strawberries, and then a couple of ornamentals: sealing wax palm and tea, and then roses,
So
orchids.
there's some for pretty what they look like, some trees, and then small vegetables. Is that what I'm hearing?
Yeah, mostly vegetables, but there's, I mean, everything. There's corn, squashes and pumpkins and Asian greens and herbs and even lavender, roses, orchids, tea, and sealing wax palm. And there was somebody asking about what about the mycorrhizal, potting mixes sometimes include mycorrhiza. So just asking if it's a fungi, but James kind of typed the response to that. And I linked to one of our publications from the Plant Nutrient Management handbook, CTAR's handbook, where from Dr. Hapte, who retired, he was our soil microbiologist, but he did a lot of work on the dependence of certain crops or native plants with mycorrhizal fungi, because they vary in their actual dependence on mycorrhiza.
The crucifers were a group that usually didn't, but most other plants, and back in Alaska, there was tree revegetation after coal mines. And what they did to inoculate the trees that they were planting, they just took a scoop of soil from an intact forest. So they would go to the edge of the strip mine or whatever and just taking a shovelful. So it wasn't even that hard, but it was very helpful, and they have data to show that.
Oh, wonderful. Yeah, here we recommend that people might look for some halekoa trees or lucina, because they're highly dependent on mycorrhiza. So usually there's the soil underneath their roots usually has a lot of mycorrhiza. And
take some from another plant that's doing well.
Mm-hmm. But one question, a couple of questions are starting to come in now.
Okay.
One was about leaf spots on avocado. And there's another on how to treat Rhizoctonia in native Bacopa. Is there a systemic fungicide that will work on Plumeria Rust?
Not that I know of.
Maybe, I don't know if Russell's here today, but he would be the agent to that. Maybe James could help with that.
No, Russell's on Maui today, Kalani.
Oh, that's right. Okay, I thought so. Okay. Let's see. There's a longer question. A tree in nature drops infected leaves and branches. Naturally, in a strong ecosystem, a predator will find the bacteria or fungus or insect and establish a colony to check and balance the problem. Would you recommend this for home garden practices or not?
One of the talks that we just had at the CBB and CLR is the abbreviations for coffee berry borer and coffee leaf rust. And we had the people in Puerto Rico saying that shortly after coffee leaf rust arose, they had other fungi that were called hyperparasites. So fungi on the fungi that were causing a disease on the plants. So actually, it's already happening in many places, and certainly here. And it's fun for people that study fungi to look at these hyperparasites. So it's hard to know the best way to encourage that, but it is certainly the case that there are other fungi that are helping minimize the effect of pathogens.
Great. I'd say that when I was studying more on insects, insect and plant ecology in a botany class, it seems like usually natural enemies or predators and things don't fully control your pest problems usually, but they can really help. But they're not necessarily dependable, and they may vary, go up and down in cycles.
Yes, actually, there was talk about that in an ecology class once, where if you had one orange and you had mites and predator mites, like spider mites, and then the predator mites on that orange, the predator mites would eat all of the spider mites and then they would starve and die. But if you took like an egg carton and you put 12 oranges, and the predator mites would eat a lot of the spider mites, but there would usually be some escapees. And so there was an actual recommendation that I heard about when I worked in Alaska that said, if you think you might be getting mites and you're buying these predator mites, they need something to eat. So actually put some spider mites in your greenhouse before you release the predator mites. And I can't do that. I would, but they're right. If you really want it to be sustainable and there's enough places for a small population to hide, that you're entrusting these predator mites to do such a good job. So finding that balance, I always say it's like riding a bicycle. That balance has to be worked on all the time. So if you have pests and you have predators or parasites of those pests, who's going to get the upper hand? And the bottom line is you want the plant to have the upper hand so that the hyperparasite or predator or whatever, but the balance is different in every place. So I'm pretty close to where I wanted to be, but it is 11 o'clock and I should be stopping talking fairly soon. So do you mind if we start on powdery mildew?
Yeah, we'll get started, and maybe one last question, and we'll try to do, we'll try to address the rest of the questions at the end of the lecture.
Yes.
But since you're going to talk about powdery mildew, someone's asking, if you have powdery mildew in your yard, is it there forever?
No. That powdery mildew, again, we're talking about what gets the upper hand, and it also depends on what type of powdery mildew, and are there any bits of structure that leave on the ground and start the cycle again. Here we don't have a winter that kills everything, so it is more likely that a little bit will stay around so that powdery mildew continues. But this micrograph on the bottom, it shows you the hypha, the little threads that are going down to rob the leaf of sugars. And then it shows the top here, but this is going to be the powdery stuff that break off and blow to the next leaf. And they also have another structure that sometimes I've seen on lilac. It forms the sexual recombination part. So with powdery mildew, you can try to make the conditions less. There are some things that I've heard of, baking soda being used at a certain level, and sometimes with or without oil. So there are home remedies sometimes that they have recommended for home gardeners. And now they've come out with some products that utilize hydrogen peroxide. Again, you have to be very careful with pesticides. Even baking soda, is it going to kill your plant? Are you using it correctly? But there are some things that could help. In Hawaii, it's not likely that it will ever completely go away. But there are some times when you remove the host plant, like if you had this powdery mildew of rose, it is Sphaerotheca pinosa, it is entirely different from these two that are Oidium species. And so what's on this papaya is not going to move to the rose. So if you remove rose for a period of time and then you bring in new rose, it's possible that by removing the host for a little while, you could remove powdery mildew. But it's probably better to manipulate the conditions so the airflow is better. And, you know, it is finding out what works best in your garden or
Thanks.
whoever's asking you.
Thanks, Roseanne. I think, yeah, that comes back to your triangle of the
Yep.
three things, right? The host, the conditions, and the pathogen. So the pathogen might come and go, but it'll mainly come with the conditions or the host. So those are things you can control, right?
Yep. That there's a lot of things that people have learned that the pathogen is not likely to keep the upper hand. It's sometimes, if you think of it as a teeter-totter and the pathogen gets to harvest more than we do, or, you know, but it doesn't stay that way. The part where you can change those conditions and make it more favorable to what you're looking for. So one of the things before we move from this slide is I wanted to mention that powdery mildews are usually very specific in what species they're going to grow on. They don't grow on a lot of things like Botrytis does. And this is also the case for rusts. We have a lot of rusts here, and coffee leaf rust is pretty much on coffee and nothing else. Plumeria rust is a very different genus and species that would not move to coffee. So that part where it's a difference between a generalist and a specialist, mildews and rusts are usually specialists. And that is a case where you can remove the host for a certain period of time and possibly help. Okay, I have to click over here. So we have another powdery mildew. This is on pepper. And again, the one on poinsettia. And again, you can see that there's two nuclei in these spores and that they're powdery and blow easily. But they don't blow that far. Rust blow further. So Phylosticta leaf spot, I have seen this a couple times already. And oftentimes, Brian was telling me more that you have an orchid with a few of these spots. And if they get low light or drought or put in a box or something like that, then all of a sudden it gets a whole lot worse until the plant drops the leaves. So I've heard that if you're growing orchids, it may become important to, once Phylosticta is blowing around in a greenhouse, that a commercial grower may spray to cover the leaf surface of the new growing point, because it's not easy to make the conditions not conducive. And if you have a plant that's looking pretty good and you put it in a box to ship it, and this is what happens, then, you know, that small change in the environment gives the pathogen the upper hand. So Phylosticta is pretty common. I think the next one, this is something that a picture I took in the lab, and these little peppers here. So it looks like peppers. Those are the Pycnidia structures or the Parathecia structures that are going to release spores to blow it over. And when Brian looked at this, he said that this stuff looks like Calototricum. And indeed it does. Calototricum is a generalist. It goes on a lot of things. It's kind of like Botrytis. It usually doesn't get the upper hand unless, you know, there's a really conducive environment or a really susceptible host. So I wanted to put these names up here so you'll have seen them once. Fusarium is that causes a lot of root rots, but it also causes other things. It's also a secondary invader, and there's a lot of different kinds, some of which don't cause problems, but some kinds do. So Fusarium is well studied by plant pathologists, and read about which type you think might be causing a problem. It has distinctive spores under the microscope, which is what I look for. Alternaria is another one that I see once in a while. It has distinctive spores. Cercospora is a fungus that I didn't ever see in Alaska, but it is very common here. Calototricum I have seen everywhere I've been, kind of like Botrytis. And just of interest to me, how I said Calototricum, say it with confidence. There was a person studying this in Puerto Rico, and she was studying different types. And she said Calototricum. And so that's another pronunciation that is well used in Puerto Rico. So it doesn't matter that it's pronounced differently in different places. If you write it on paper, it's the same one. One time I was reading a paper from Russia, and this is what would be written. Rhizoctonia solani was written down in the paper, and I knew what they were talking about. Phytophthora infestans was the late blight organism, and I knew what they were talking about. But the names of the problems on a plant can be whatever. It's like a common name of a flower. A person can choose a different one. If it's a leaf spot, can be small or big. A blight, something's wrong with it. A wilt usually implies that the stem is a problem. A canker is usually on a stem in a very small area. And then in general, rot just means that the tissue decomposed. And going back to this Rapidohea death that we have found here, that was literally brought to Brian. Brian talked to other people, and they named it. And it'll be shown a little bit later. They later realized that there's 2 different species of the same genus causing it, and they were actually new to science. They were described. So that part where this may not have been imported, it may just have somehow gained a foothold. But rod is very common, and there has been a very good citizen science movement saying, please clean your feet when you move between these places. And when you are at a hula contest, please don't take stuff from the native forest. Don't transport ohia, and even firewood. Burn it where you buy it, where you burn it. Use it where you got it. Do not carry branches that may carry this from one place to another. So Calototricum gliosporoides is how I say it, instead of Calototricum. But anthracnose is the name of the symptom, and that usually means it's on the surface. There was another symptom caused by this that was internal rot of coffee berries, and they decided to call it coffee fruit rot because anthracnose is only for on the surface of leaves or fruit. Down here are the little spores that blow. And Calototricum, it is an opportunistic pathogen. It is blowing around, but it doesn't always get started unless the plant is stressed for some reason. Plumeria Rust. And I heard a question during the break, like, what is the best control for this? Is there a systemic available? The answer is I don't know. Here's the little spores, but this Plumeria Rust is an entirely different genus and species from coffee leaf rust. Rusts are usually very specific to certain plants. So if you have different kinds of plants, the rust is not likely to move from one to the other. And I would advise Google searching for more information on how to control this. I wanted to show you coffee leaf rust. This was a picture taken on Kona. the plant, starts out like this. And then here you can see just a few spots, but this is probably like 2 months later. When 20% of the leaf is covered, then the plant drops its leaves. So coffee berry borer might destroy the coffee berry, but it doesn't kill the plant. Coffee leaf rust, if you have a plant that drops all its leaves, then a defoliated plant can die and you affect next year's crop. So coffee leaf rust is a hard new disease to deal with. And it's probably spraying, there's some coppers, there's some bacillus, they're looking at a section 18 for a systemic. And just that, on the one hand, it's sad that we have this. On the other hand, as soon as it was identified, there was all this information about what is the life cycle and when can you control it. So people have a lot of information to work with. It still takes time and money, but the information was there quickly. And we have to find out if what works in Colombia or Mexico or Brazil, if some of those things work here, and the best time and best way to apply it. The one thing that I would say is we're living through the coronavirus. I've had to learn to use Zoom because of it. But again, the best time to treat coffee leaf rust is below 5% incidence. So if you can find it on 5% of the leaves in the field, just a little bit, that's when you have to spray to cover the leaf surface with things that won't let it germinate. Because once it germinates, those sprays are not going to kill that lesion. And there's literally, it will blow on the wind to the rest of the field and maybe other places. One of the first places they found this was on wild coffee. So coffee that was not being managed. So coffee leaf rust will stay around even when you don't have coffee, because it can blow on the wind and live on wild coffee. And then this was a silly picture where I tried to use my cell phone to show this little long thing that was particular for this fungus. But this is what I look at to finish the determination of what it might be. And Basil downy mildew, I'm going to let you see what it looks like and say that you saw it. But as I said earlier, it seems like basil is very susceptible to a lot of things. So growing it, the best thing you can do is exclude it. Once it gets started, sometimes it's better to switch to a resistant variety or stop growing basil. This is called Peronospora. And cold, wet weather. So we have some places that are higher up there like that. Contaminated seed. So sometimes it isn't in the area till you bring it in unknowingly. And sometimes if you know the seed might be contaminated, there's hot water treatments that would prevent that, but it also sometimes lowers the germination rate. So all of these things are possible ways to reduce the impact of a fungus. Taro leaf blight. Now again, we have Phytophthora here. These sporangia, usually when they are, sort of lemon or egg shaped, but when they get in water, they empty out these little swimming spores, which swim to other places. So in the rain, this can spread over to here, sometimes on the leaf margin, because in the morning, that's where the dew collects. So Phytophthora, you're looking about how does it spread in water? And then late blight is actually called famine on the wind. But usually Phytophtheras and Pythiums are how do they spread by water moving from one plant to another. We have now finished fungal diseases, which is 85%. So bacterial diseases, nematode diseases, viral diseases, there's about 3 slides each because we want to stop for questions pretty soon. So there we go. Look at all of those. bacterial spot, bacterial speck, bacterial canker. This Erwinia chrysanthemi, there's also something that used to be called Erwinia carotabra, and that's called soft rot. And it degrades the pectin in the cell walls and makes it mush. It also makes it smell really bad. And I almost guess Erwinia sometimes by the smell. So here is Xanthomonas. It's bright yellow in colonies, but again, it's usually going to be spread by water splashes or equipment. So it is easier to control stuff like that if you have an understanding of bacteria can't fly through the air. They usually have to be moved either mechanically or water splash. Pseudomonas is another. Chicoria probably is talking about lettuce. And, you know, it will make this plant unusable and usually limiting the conditions. I want to mention this one here, Ralstonia solanacearum, because this is one plant pathologists dread. Once it's in the soil to stay. So if you have a seed source that you're not sure about for something, you know, if there's the potential you might be carrying this onto your farm, you know, you think twice. When it's in the soil, it's in the soil. It bothers bananas, tomatoes, potatoes, you know, ginger. This is one that I would like to never see in my lifetime, but it is very much in patches on the island. And one of the things that people that grow ginger, they have started to do it in pots above ground, so that if it's in the soil, then the plant no longer gets to touch the soil and spread through water. And this is also a case of where, you know, like Rapidohea death, think about your shoes, think about the tires of vehicles. You don't want to spread this if you have an inkling that it might be there. So bacterial streaming. So this is the stem of something, probably could be Ralstonia. And the little cells you can see as these milky swirls coming out of it. You know, with a fungus, you wouldn't see this. And all this tells you is there's a lot of cells that are clogging up the vascular system that's supposed to be transporting sugar and nutrients back and forth between leaves and roots. So if you see this, you want to dispose of it where it won't get in any other plants. And usually there's culturing techniques or PCR molecular techniques that would show you exactly what it is. But for a home garden, you just take this plant and you put it in the trash. I don't even know if you would compost it. There we go. Bacterial diseases all done. Nematodes. So these pictures show what the little worm looks like. This shows what it can do to plants. With coffee plants, there's a coffee root knot nematode, and they said after about 10 years, if the plant is wobbly, you can literally pull the coffee plant out of the soil because the roots have been destroyed by certain nematodes. Nematodes are very common in the tropics. So if you get plant parasitic nematodes, it is hard to know the best way. It's usually a lot of work. For the coffee root knot nematodes, they say start clean, stay clean. But if you're not clean, then you actually replant your coffee farm with grafted trees. You graft the kind of coffee you want onto resistant rootstock. So going back over here, they're very small. You see them under a microscope. Brian was very good at identifying them. I don't work enough hours to do that. So now if it's nematodes, the sample will go to Oahu. 6,000 species known. Indistinct and usually mimic nutrient deficiency. So you can tell that this plant isn't doing well and the roots are often very rotted, but is that because of a nematode or something else? Usually it would take a microscope to figure that out. And root knot nematodes might be specific, like rust and mildew, but then there are other nematodes that just pierce the plant cells and they attack all kinds, like Botrytis is a fungus that does a similar thing. So soil assays for accurate diagnosis. The management strategies directed at the particular nematode species present. But if you're in a garden and you grow things in containers, then that part of just make an air gap so that soil and water that is running on the ground doesn't get into every pot. I just listened to burrowing nematodes in anthuriums and how much work it is, because once the burrowing nematode is present, then pretty much it moves wherever the water moves. So this is showing you some root knot nematodes that grow actually, the entire nematode is inside the root. And in this place, the nematode is sticking out, but just the little mouth part is sticking in. And again, a microscope is what's going to show you this. But the symptoms, it's patchwork, because they don't swim that far. It's where do they get the upper hand? You know, the amount of roots translates into the amount of tops, very stunted versus okay. And here it's a patch in corn. Again, we have a lot of nematodes, and you watch how you transfer soil from one place to another. Here is Meloidogyne incognita. The root knot nematode on Kona coffee is Meloidogyne conensis, and it is specific to coffee, whereas this one is more common on a lot of things. But these roots with these big knots in them where the nematodes are not going to get water and nutrients as well as this healthy root system. So again, there are not as many nematodes as fungus. Usually the nematodes are only on the roots. And here's an exception. Apellincoides frigerii is how I'll say it. And it is a case of a foliar nematode. So these, we saw them in the very first, they're stunted because a nematode is above ground. And so if you have access to a microscope or a hand lens, you might possibly notice that. Usually a hand lens is going to tell you if there's fungal spores that you can see, and if I get a sample like this, I would take something from the edge of healthy tissue and diseased tissue, and I might put this on a plate of agar. And in this case, I would get, I might get secondary fungus, but I would see little nematodes swimming around on top of the agar. So some of these, they are not that common, but if you have them, the plant is unhappy. So this is some more places where nematodes have greatly affected the crops. And in the old days, you used to fumigate the crop or fumigate the soil, or sometimes now you could use steam, but it's hard to get nematodes out of the soil. What I've heard for coffee is when they're there, and now you have to use grafted on resistant rootstocks. So the first thing, like I said, was try to exclude this from your gardens. And then if you can't exclude it, then you go to the next steps. The last bit, viral diseases. So viral diseases are often, they have to be spread in some manner. They can't move on their own. Aphids are a big way that this happens. This clip, I don't have it actually working, but it's talking about Non-persistent versus persistent. Non-persistent is where it's on the aphid mouth part and it's just sticking there on the surface. So if it goes to a plant with a virus, the next couple leaves it goes to will get a little bit of virus particles and then it's usually washed off. But if it's persistent, the aphid gets the virus in its gut and its salivary glands. And then for the rest of its life, if it's persistent, this aphid will pass it on to whatever plant it's feeding on. So in some respects, it's easier to stop this than it is to stop this, because a persistent vector is when the aphid has to be killed because it will spread the virus for the rest of its life. I wanted to show you how I test for viruses. I have a list of about 20 things. These are an immunostrip where you have a small bag, you grind up a certain amount of plant materials. That's why it looks green. We actually have a drill press in the lab to do this. And then you follow the directions, put this line in the liquid, and the liquid moves up. And this is the control line. That means that the strip is working. This is the Cymbidium mosaic bias test line, which says that this sample had that virus present. And, you know, as Brian would say, it's lit up like a Christmas tree. This particular little piece of tissue had a lot of that virus particle. However, it did not have the ORSV virus. On this case, you could have seen both. So if somebody asks if it's this virus, this test can be done in an hour or less. And they actually make these strips and sell them to growers. If the growers want to do them themselves, it's less than $12 a strip. But usually I'm only going to test for a virus if the agent tells me to, because these strips cost money as well. And it's usually used with intent. This I'm going to scroll through quickly because I think you've already heard of a lot of integrated pest management already. And if not, the agents can ask me again. But what we started with is keep them out. Regulatory control is a quarantine. Cultural control, crop rotation, weed management, just make the conditions less. Host resistance, biological control. They're talking about mechanical control in Florida, growing some citrus inside of screen houses to keep out citrus greening. You know, that takes a lot of work, but at this point, the damage from citrus greening is that great. So we mentioned some of the soft or natural pesticides, you know, baking soda, soil or soaps, oil. There are things that can be done on a garden scale that are minimalism instead of chemical control that you use if needed. I spray Talstar around the base of my house because I don't want fire ants. And I have seen them all over the doorstep until I started using this. So the part about chemical control, you are using the label is the law, and there's a certain point, but it's usually after you've tried everything else. And there's lots of information saying what the latest is. So this is a repeat: physical methods, biological methods, chemical methods. This is Brian's ohia rust. You know, this has not been here all the time, but I personally was given a small ohia seedling, and my son actually won it in the volcano trail race. And he said, "Mom, how's it doing?" I'm like, well, Well, dear, I'm a pathologist and I know what killed it, but I could not save it because I planted it in a place that didn't have enough air circulation. I have some seedlings are more susceptible than others. You can see this plant, mountain apple, I believe. Anyhow, that is really hard on Oahu. It's killing those trees. Some of the ohia seedlings are more resistant. Some of them have better conditions, but it's a rust and it's very specific to this. Cercospora leaf spot is not very specific. It's found on a lot of things. And if you see something like this, the recommendation is fertilize and clear the weeds. It is usually that cultural control. And then Plumeria Rust, the person asked, and my answer was, I don't know. So lifelong learning. There's a lot of places to look up that relevant information and hopefully get to the point of wisdom rather than just knowledge. Here's the one you don't want, bacterial wilt. Please use the quarantine as long as you can. I'm stopping right here to say that rapid ohia death, ceratocystis, it's not called fimbriata anymore. There's actually 2 Hawaiian names, and there is one Hawaiian name that is more like a plant destroyer, and there's one that is a little less virulent. It doesn't cause as much disease, but it keeps spreading both on the Big Island and to other islands. And since ohia makes up 80% of the endemic forests, this makes me really sad. Powdery mildews, more pictures. I guess that some of you haven't seen as much rapid ohia death as I have, but I'm living at ground zero, just like coqui frogs and fire ants. I wanted to finish with what I started with. The first thing you do is listen to all the quarantines. Please don't add anything by shipping something that you shouldn't do it. If you have something for testing, it goes to an extension office. This, I'm actually going to stop sharing and go to questions.
Thank you for all of that. I know we had a question in the chat and also one in person here about composting things that have diseases, such as tomatoes or papayas that have powdery mildew, and your thoughts on that.
So my thoughts on composting is if the composting is working well, it is going to take care of the pathogen. But if it's a half-hearted attempt at composting, then you might be spreading more of the pathogen. So they have composting facilities on Big Island, and little fire ant was in them. And so there were places where they wondered if they got little fire ant from the compost. Then they started being much more careful and turning the pile better. And now I would use their compost. So it matters that you do it right and that you get all the corners. It is possible, but it's not guaranteed if you're not careful with composting.
Thank you.
So I see there's some comments about basil. That's good. Thank you guys for listening to me. As you can tell, I like to talk about plant diseases. And it's not often that people want to listen. The part about, I think I said it at the beginning to Laura, that a good plant pathologist is a person that the diseases are not a problem. You have given people enough information that they know how to control it. And sometimes though, they come to you with a plant such as banana bunchy top, and the answer is chop the plant out. It's very sad that the treatment is removal of the plant.
Roseanne, we
But
had
sometimes your neighbors will thank you.
we had a question. We had a couple questions in Hilo. One of them is on your slide with fungus, about fungus. You had it listed about blight, cankers. Are those symptoms or are those different kind of funguses?
Those are symptoms. There's actually big charts saying all the different names that you can call the disease symptoms. When they talk about a disease, they'll usually say symptoms, which are the plant response, and then the sign is the actual fungus. So if you had botrytis, the sign is the gray fuzz on there, or the powdery mildew, the white fuzz. But the symptom is that the plant leaf is yellow, chlorotic, and that the strawberry fruit is mushy. So a sign is an actual part of the fungus or the pathogen, and then a symptom is the plant reaction that's usually showing it's been harmed.
Awesome. And the other question we had was, it's more of a situation. So a plant, heliconia, was growing since the 1950s in one spot, and then all of a sudden, it just is starting to fall over. So how would you go through and basically helping that homeowner just figure out what is happening to this plant?
That's a good question, and I don't have an easy answer off the top of my head. But when we started asking if something has changed, sometimes when a plant gets really old, that there is, and so I'm kind of giving another example. There's called apple replant disease, where the apple, a tree, has so many feeder roots and they have so much fungus growing on them that when you take out that apple tree and you try to plant a new one into it, that fungus jumps on the new one and makes the replanted tree do poorly or even die. And so the heliconia that has been there a very long time, there's probably been other things down on the roots. And it's possible that some of them may have either got the upper hand or something was introduced. So it's like, how can you change the tide to favor the plant roots again? Or take cuttings and put it in another place. But it'll be a long time before the flowers are as beautiful.
We have a question on this end about baking potting soil or steaming, other ways of sanitizing potting media. Do you have a preferred or suggested method?
When I was going to school, they had this very large autoclave and they steamed the soil and they steamed the pots. And, it was a lot of time and effort and care to do that. So it's possible if you have the right setup. They were actually, when they talked about burrowing nematode and anthuriums, there was a way that they were steaming the cinders in a large truck. The details of how to do that, I don't know off the top of my head. But I do know that big and small, it is a choice. It's probably not good to try it in your own oven. You would want to do it outside in a different way.
Thank you for that. I also see a question in the chat. Unfortunately, I think the person left, but it's a question I know as an agent I get a lot. Folks are asking about using milk as an antifungal thing, and supposedly the lactic acid affects the fungi. Do you know if there's any evidence for or against this?
So off the top of my head, I can say that I don't know about that. I have also heard about like Korean natural farming methods here on Big Island, and I haven't had time to look into that. So there are some of these things that with fine tuning have the possibility of being helpful. But compost tea was very popular a while back in Alaska for home gardenings. And compost tea, it mattered if it was fresh, you know, and if it had the right starter in it. So sometimes compost tea seemed to show an effect and sometimes it didn't. So I don't know how milk would fall into that category.
Thank you for that.
I heard some of the plants that everybody likes to grow, you know, some that you can eat, some that you can look at, some that are large that take a long time. And I encourage you all to make your own little paradise and help others make theirs. I had talked to Laura at the very beginning, say, if you want a plant pathology example, it's like if I'm the tree, you know, and I have roots, you know, I have 40 years of plant pathology experience, but you guys are the mycorrhizae. You touch far more people than one person can touch. And if it's a particularly troublesome or quote, interesting, you would send it in to the ADSC and you would have somebody that has a microscope look at it. But we appreciate that you're out there with eyes and ears listening and enforcing the importance of, you know, all of the regulations to keep things out as long as possible. We are an island.
I see a question in the chat that says, how long do you wait after digging out the banana mat trunks and roots before you can replant banana keikis in the same area?
That's a very good question. Like I said at the beginning, good questions. I don't know off the top of my head, but I believe that there's probably some good recommendations out there. So that, ask your agent to help you look that up.
I see another question in the chat about, I'm going to sort of broaden it a little bit. They were asking about using kind of microbial preparations like EM1 or EM2 in compost to decrease diseases. Do you have any thoughts about that in terms of like beneficial microbes or nematodes or anything that might help?
So actually during the break, we were talking about mycorrhizae and potting soil and things. And I was saying that in some places for replanting, this was after, it was a friend who did a study on replanting trees after coal mine stripping. So you're putting trees into something that has very little microbial action. And it was just like walk over to the next forest that's closest to that place and take a shovelful and put it on top. So that doesn't sound very scientific, but my friend found data to say that scooping soil from a nearby patch was helpful. The seedlings grew bigger and faster. So all of these things, with which microbes and how many, it's hard for scientists to quantify easily. You know, they sometimes call it the black box, but the soil is very complex, and the more organic matter and microbes that are in there, in the long run, that's going to give it a resiliency. They were looking at it for Kona coffee. If you could use compost, that it would stimulate things that would keep the nematodes down. And under some conditions, yes, but exactly what those conditions are. So it's hard to, there's spotty results. Sometimes there's a significant benefit, sometimes there's not. And it's hard to say why.
Awesome answer. Thank you for that. And thank you for also sharing the conversation during the break. I know not everyone was able to hear it. So that's great.
Again, I want to thank you all for being master gardeners and both listening to all the instruction and then sharing it with 40 hours of volunteer and hopefully more than 40 hours. But I have seen master gardeners in Alaska previously, and I look forward to working with you here. Although, as I said, as Brian Bush, who was the previous diagnostician, worked 40 hours a week and always had something to do. And I'm only working 10 hours a week. So a lot of times I actually give as many questions to the master gardeners as I can, because I have to keep my 10 hours and get other work done too. I'm also retired and enjoying it. And if you move to Hawaii when you're retired, you want to have some beach time. So
Thank you for all that you do and for giving this talk. I know I learned a lot, and I think everyone else did too. And we'll definitely try to take some work off your plate, but we'll certainly be bugging you with more questions in the future.
questions are the lifeblood of science. And now it's just sifting through all that information that's out there. And sometimes, go back with a second guess, or literally the next best thing to being right is being wrong. If I say, oh, it looks like this plant got too much water, and they say, oh no, it's by a driveway. And then it's like, oh, maybe the roots were compact. And so it had the same effect, that the roots were not as vigorous as they needed to be. So that part about asking more questions and being willing to change your ideas, that is a hallmark of science. And it's a good thing to be flexible rather than rigid in what you think's happening.
Very true. Thank you for that. And I think with that, I know Kalani posted a link to Oahu in the chat. I posted one to Hawaii. Thank you again. I think we're going to go off to our individual island groups, but this was really great.
Well,
yes, thank you, Roseanne. Thanks a lot.
You're welcome.
Awesome job. And you gave us a segue into next week's class where we'll actually try and practice diagnosing these problems.
Teamwork, teamwork. All right, bye.
Take care. Bye.
Bye, Roseanne. Hopefully we'll get to see. Oh, she clicked off. Bye.